Welcome to the Supplement Engineer podcast. My name is Robert Chinesky. Joining us today is gut health expert, certified sports nutritionist, and a board member for the Nutrition Coaching Global Mastermind, Dr. Gabrielle Fundaro. Thank you for joining us, Dr. Fundaro. How are you doing?
Thank you for having me, and I am doing well. I'm in the midst of some actually super gloomy weather for Arizona, which is very strange.
Yeah, we almost be part of the same weather band we are, because in Austin here, it's been overcast all yesterday, all today, and I think even through the rest of the end of the week. It's just super gloomy and kind of rainy and misty and everything. And so
yeah, I know. It's okay. It's one of 5 days of the year that I have to deal with clouds.
Awesome. Well, thank you for joining us today. You've been on a number of podcasts, so I don't want to spend too much time going through your background, but for listeners, anytime I have a first-time guest on the show, I like to get at least some of the background. So if you could go over, the high points of your background, and, we can get into the meat of the interview from here.
Yeah, sure. So my bachelor's is in exercise, sport, and health ed. And I, in my junior year, switched from wanting to run a gym to wanting to teach at the collegiate level. So I decided, okay, I'm going to go on for my PhD. And I did that at Virginia Tech. So I actually ended up studying serendipitously the relationship between the gut microbiome and human metabolism. And from there, I went on to teach in exercise science, primarily sport nutrition and anatomy and phys, and really pursued more of my interests around sport nutrition. And so that's when I earned my Issn sport nutritionist certification. After about 3 years of teaching, I was found by Mike Israetel of Renaissance Periodization, and I was recruited as an RP to make a decision about the trajectory of the rest of my career. And I found coaching to be incredibly fulfilling and really an outlet for me to continue to grow as a practitioner and an educator, just in different ways. So I was speaking infrequently about the gut microbiome and sport nutrition at that time. After that year, I resigned and went to coaching full time. And within that, my first year of coaching full time, I started my own business as well, Vitamin PhD Nutrition Telehealth, to serve a different group of clientele. And I had the amazing opportunity of speaking locally and internationally about the gut microbiome and its relationship to human metabolism and exercise performance. And I've created plenty of content around that. And because I wanted to improve and continue to grow as a coach, I pursued health coach certification and education in motivational interviewing, cognitive behavioral coaching, acceptance and commitment therapy interventions. And over a year ago now, I started the Bridging the Gap article series with my colleague Shannon Beer, where we talk about sort of the historical, philosophical, and cultural aspects of the fitness and wellness industries. And we have since then, with the help of some sport and health psychologists and a member of MINT, the Motivational Interviewing Network of Trainers, developed a coaching framework. That's sort of it's nothing that's new. It's not newfangled. It's just that we've taken some of the best practices and the foundation of motivational interviewing and kind of built on that a structure. Kind of one of my colleagues has put it in a way that I think is helpful, that it's operationalized. And so that's what I'm up to now. And it was actually Mike's idea for me to even speak about gut health and the gut microbiome. It was sort of a means to an end. I thought, well, I need to get a PhD. And, I was studying skeletal muscle hypertrophy initially, and we just had some accidents with that study. And so my side study on probiotics became then my dissertation project. And he was like, yeah, it seems like it's a hot topic, burgeoning. And this was back in 2017. And I was like, I guess I can talk about it. I mean, if people want to hear about that. And it went from being a very niche area back in 2014 when I finished to now, I mean, it's, obviously one of kind of the hottest topics. So I feel fortunate that, as luck would have it, that happened to be what I studied. And so I try to provide, an evidence-based perspective that's also practical for people.
Excellent. Yeah, I was curious because, you got your PhD in 2014. So I was curious, now gut health is mainstream. Everybody wants to know about prebiotics, postbiotics, probiotics, all of those things. And I was trying to think, 6, 7, 8 years ago, because, getting a PhD is a multi-year process. It's not like you just sign up and then you're done in 6 months. So I'm curious, what was it specifically that attracted you to like the fascinating macrocosm that is the gut?
So the lab that I was working in was a skeletal muscle phys and biochem lab. And we were studying the downstream effects of a specific immune receptor activation and how that might play into metabolic dysregulation.
Okay.
And so what we would do with our mouse samples or our cell culture samples, we would treat them with something called LPS or lipopolysaccharide. And when we were using our human subjects, we would often do a high fat feeding challenge. So we would feed them a super high fat meal like Jimmy Dean's breakfast sandwiches. It was okay that I say that brand, but that's what it was. And we would measure plasma LPS or circulating LPS in a time course after the meal. And we found that it was acutely elevated and then, would come back to baseline. And I was wondering, why is it that we're looking at LPS? Obviously, we were treating mice with this, and what is the physiological relevance? So LPS is actually a constituent of the cell wall of certain types of bacteria. And when those bacteria die off or are destroyed, that becomes liberated, and it can actually sort of leak out of the GI tract, perhaps between the intestinal cells, or maybe carried out by some of the packaging that we use to transport fats out of the intestine. Okay. And it can bind to this immune receptor that's located in all sorts of tissues, but we were looking at skeletal muscle specifically. And from there, it causes this cascade of inflammation that can induce insulin resistance and also reduce our capacity to fully break down fatty acids. And then that can lead to sort of these incomplete, just sort of leftovers that can actually serve as inflammatory signaling molecules as well. And so I thought, well, if it's coming from the gut, why don't we look at that? Because that seems like an area for a solution. Right. And it was not our area. And so there wasn't really an opportunity for me to study that. And then lo and behold, maybe about a year later, the company that produces VSL3 was providing some funding for research looking at, the potential protective effects of probiotics for a bunch of different applications. And so we ended up writing a grant to study probiotic supplementation as a potential protective mechanism against this high fat feeding induced metabolic dysregulation. And it was really just, the main study that I had was looking at high fat feeding and hypertrophy. And we just lost those samples because of just some improper storage, and the marker came off and it was like, we can't use that anymore, but thank goodness I have, all this probiotic stuff going on. And so, it was just a matter of curiosity. And I really had no idea. I didn't know that the gut microbiome existed before that. I was just like, oh, wow. Okay. So we've got a lot of bacteria in there. I mean, yeah, that makes sense. Cause poop is like dangerous. But I didn't realize, its relationship with like really all of human physiology. And at that point, metabolic endotoxemia, that phenomenon of that low grade, circulating inflammation, I think it had been coined in not long before. So this was in 2009 when I started. So it was a 5-year process to finish. And that was a relatively new term. And I just remember thinking, oh my gosh, there's this whole world that we don't talk about, we don't think about. It was nothing that people were speaking about in terms of sport performance or metabolic health. And even some of the ideas that we had back then, like enterotypes, which are sort of like somatotypes, like ectomorph, mesomorph, have been pretty much shown to be inaccurate, and not applicable. But at the time it was like, oh, I think there are these enterotypes, there are some like obesogenic microbiome profiles. And now we know that's not really the case because technology has improved so much. But yeah, so that was kind of the start of it. And I didn't do anything with it between, 2014 to 2017, except, I tried to, get some publications out and whatnot. But I was like, I don't think I'm going to do anything with this ever again. No one's talking about this stuff. No one cares about it. Lo and behold.
Since you were involved with, writing grants and getting studies off the ground, and one thing I always like to do on the podcast is kind of like peel back the research layer, because from the lay people side, we read the studies, we see the abstracts, we can get access to some of the full journals. But what I like to do with the podcast is also give the listeners kind of like a behind the curtain look at what goes on into doing that, because people always complain, why don't we see more studies on this ingredient? Why don't we see more studies on this? And I'm thinking, well, it's not like the FDA or the NIH is going to be giving money to supplement companies to figure out how to get more gains, build up your boost, your bench press or anything. So from somebody, can you speak to like all the different steps that you have to take just to even secure funding for some of these studies, and what's the cost, expense, and the time commitment that's involved with those?
Oh yeah. Huge time commitment. And it really depends too on the institution that you're kind of like operating within, and how new of a researcher you are. So there are grants that are made for new researchers. So it's people who have just finished their PhDs, they're new faculty, and it may be a little bit easier. The threshold is a little bit lower for them to actually receive that money. Whereas someone who's at a really advanced institution, like at Virginia Tech, you have immense competition for a limited pool of money. And you also kind of have categories. So they are looking to fund certain types of research. So you won't necessarily get funding for your super cool idea if no one is looking to fund it.
Right.
And then you can also have like an industry funded study. So there might be a company that says, we want you to study the effects of this supplement. We're going to give it to you. We still want it to be, double blind randomized control trial. They're not going to like necessarily influence the results, but they may have some say in when those results are published, and perhaps if they want to review them first. And of course, there's also a positive publishing bias. So once you have gone through the process of, okay, you've written this grant, which takes, weeks with a team of people, because you have to do a review of the literature. So you have to look and see what is the state of literature right now. So you're basically like writing a little bit of a systematic review. And then you have to come up with your budget, everything that you're going to need and how much all of that costs. And then the design of the study. So you kind of already have to have everything conceptualized ahead of time. And then you send it in, and then you may or may not be approved for, some or all of the money that you've asked for. Right. And then if you only get a portion of that money, now you have to decide, okay, what's the stuff that we're going to not do because we don't have enough to support, all of the assays that we want to run. And then you go through and, you do the study, and like, it's science. And so it's not about like proving things. It's just about being a little less wrong over time, and, learning as you go, and like samples, aren't stored right, or, the assay doesn't work, or whatever it is. When you're working with humans, there's subject dropout. It's really difficult to have, tight controls on in human subjects. And then you spend months, writing after all of the data analysis is done. And then you might have to pay for publication fees as well. So there might be several $1,000s for you to publish in an open access journal if you want people to read it, or you publish behind a paywall. And then, I think there's like it might've been Eric, was it Eric Trexler or Eric Helms? One of the Erics, I think, said that like there's an average of like, or maybe Andy Galpin, but like 7 people will read a published article, like that's the average. Yep. So, it has to be a labor of love. And when you're in a really niche area, so I actually just helped write a grant with a faculty member out of Lipscomb University in Tennessee. So Jeremy Townsend, I met him a couple years ago now at an ISSN conference, and we designed a study looking at GI distress and how it might be associated with gender-specific differences in the microbiome after resistance training. And this is a really new area. There's really there's one other study looking at GI function after an intense resistance training bout. And it was difficult for us to secure funding because it's just not something that people are interested in. It's like, well, it's kind of sport performance, but then you're looking at like NSCA or something, and they're kind of like, they want to look at like more of the performance aspects. And then we're like, well, it's not really necessarily a health concern because it's GI distress, after resistance training. Yeah. So finding funding for your really cool idea might be difficult, even if it's an awesome, like new area. To some extent, we see kind of these trends in what people are looking at, especially with like probiotic strains, because so many other people have already done the work there. You have a strong foundation to say like, hey, here's all this other data, but there's this one question that's not answered, and there's enough interest in it now that we can procure funding for that.
Okay, great. I'm glad you brought up the lack of the lack of reach that a lot of the research has, and in fact that most of them don't. Because I've had Dr. Trexler on the show before, and I've listened to a bunch of his Stronger by Science podcasts, and there's so many. I don't even know. There was something him and I touched. Like, how do you bridge the gap between like with the researcher and the layperson, but then also just kind of like opening up access to things, making it less so highbrow and talking more in like a conventional terms or something like that. But there's a bunch of not problem, like the system's not broken. I don't want to say that, but like the researchers aren't getting paid from behind these paywall things. The peer reviewers aren't getting and it's all voluntary. And my younger sister is a pharmacist and she does peer review for a couple of medical journals and whatnot. She said, "Oh, well, I've got to work a 12-hour shift at the hospital." Then I said I would do these 2 reviews. I'm just thinking, man, there seems like there are so many holes in this process. Like there's got to be a better mousetrap to do this.
100%. Yeah. Yeah. And I've been a part of a peer review process, and it's really interesting also to see like that you'll have kind of dissenting opinions. I mean, like 2 like wildly different evaluations of the same paper, and it's always like, people always joke about like, oh, reviewer number 3, so yeah, it's a time consuming process and it's certainly not infallible. And I remember when I had submitted a publication, one of the reviewers said, well, if you didn't get any results, why do you want to publish this? And like, we need to publish those null findings. We need to say like, hey, we gave these people some probiotics that didn't do anything like that. We need to see that. And it's sort of a joke that I think there's a journal, like the Journal of like Null Findings or something. So it's a journal full of things where people are like, hey, we did this thing, we didn't see any differerences. And like, that shouldn't be just a joke, that should be like, yes, we actually publish those things. And we don't necessarily see a lot of like the funnel plots that will let us know if there's a lot of bias in one area. And that's really important to consider, especially when we're looking at, the effects of like probiotics. You can find RCTs that do show that there's an effect. But then, when we want to look at kind of a larger like top-down synthesis, might look at like a meta-analysis or a systematic review, that can help. But even that, like you said, it can be so technical that it can be really difficult for people to make sense of it. Right. And, so yeah, once it's like the science communication aspect is sort of, I think, the missing like leg on the stool here, that like, even if we have really, well-designed studies and quality research, and we have people that are really hungry for that knowledge, if we can't communicate it to them in a way that makes sense, then they may instead go for the stuff that's kind of nonsense, but makes more sense.
Correct. Yeah, well put. Let's start diving into all things microbiome. So I know there's not like a textbook definition of gut health, but can you kind of describe what this amorphous term that everybody's throwing around as the buzzword, what gut health is, and then I guess briefly touch on the differences between prebiotics, postbiotics, synbiotics. There's one other one. Probiotic. Yeah, we go. Yeah. Pre, post, pro, and then syn. Yeah, if we can touch on all those, and we'll go from there.
Sure. Yeah, all the otics. Yeah. So yeah, like you said, there's not really a textbook definition of gut health. And so I've tried to create one from what I've just gleaned from lots of reading and writing. And I think when people when they say gut health, I think they mean 3 different things. I think they mean a diverse microbiome. So a richness and an evenness of all of the microbes that inhabit the GI tract. So we have relatively higher levels of the beneficial or neutral groups, and then relatively lower levels of the potential pathogens. So that's one aspect, microbial diversity. I think the other aspect is the absence of a functional or organic disease of the gut. So we don't have any diseases of the anatomy or the physiology of the gut. And I think the third thing is that they feel that they have comfortable digestion. So they're not having excess flatulence or really loose stools or constipation, that they feel that they're having comfortable bowel movements and like a normal amount of gas and bloating, based on what we're eating. And so those are what I think that people mean when they say gut health. But there's also this sort of domino effect. So when they say gut health, they're also thinking of gut health, those 3 things, and how that affects everything else in my body. And then, so the other part of your question. And not to say that definition is true. I mean, because like you said, gut health is sort of a marketing term, but that's what I think people mean. So when we're talking about all of the otics, so I think I'll start with prebiotics, then we can kind of like go, it makes sense to progress in that way. So a prebiotic is an energy source that will feed beneficial bacteria in the gut. And so most of the time we're talking about some source of fermentable fiber, what we call a microbe accessible carbohydrate, or a MAC. So that's a prebiotic. It's not something that we have to necessarily take as a supplement. It's something that we could find in fruits, vegetables, whole grains, legumes. So it's pretty ubiquitous in the diet if we're eating plants. A probiotic is a live, this is the definition of it currently, is that it's a live microorganism that we ingest for the purpose of a health benefit to the host. So it does have to be an organism that has been shown to confer some benefit to a host. So it's not just any old microbe that we would get from like a ferment, like a yogurt or something, because not all of those microorganisms are going to have shown that they do something beneficial for us. The term symbiotic is a mix of both a prebiotic, so it's some sort of a fiber, or a microbe accessible carbohydrate or energy source, and a live microorganism. And a symbiotic, the definition has been kind of developing over time because it's fairly new, but the fiber in the symbiotic should be specific to that microbe. So if you put a microbe in with a carbohydrate that it doesn't really want to ferment, that may not necessarily be considered a symbiotic. So that's just kind of like a nuance to that definition. And then a postbiotic is a product that is made via the metabolic processes of a microbe. Now, the microbe doesn't necessarily have to be a probiotic form of microbe. It can be just one of our native inhabitants. And as they are fermenting carbohydrates, they may produce gas, obviously, and then they can also produce short chain fatty acids, and those have shown to be beneficial to us as hosts in many cases. So it's just something that they have produced, and then that can be of benefit to us as well.
Okay, perfect. Do you have any examples of what like a symbiotic is? So like pre and probiotics are thrown around a lot. Postbiotic, we're starting to see a little bit more because some companies are starting to release these tributyrin-based compounds that we're starting to see. But symbiotic isn't as much in the vernacular of the everyday individual. So is there any foods like off the top of your head that you might know that would be like something we kind of encounter on a semi-regular basis?
In this case, actually, a symbiotic would be something that would be supplemented. I don't know any, I mean, I usually don't like bring up brands off the top of my head. I know one, but I don't want to name it.
Yeah, that's fine.
But I know that there is at least one on the market that contains beneficial bacteria and a carbohydrate source that would be accessible to them. But those are so new that we don't have solid data on whether they're actually any more beneficial than just taking a probiotic. And even, the efficacy of those is questionable in many cases also.
Okay. When probiotics first kind of came on the market, it was kind of like everybody needs to take this. We all need to be concerned about gut health. Now from like the research side, we're seeing that maybe not everybody needs one. It's more of like a case dependent thing. It might even be more of a strain specific thing. So can you speak to a little bit about the utility and efficacy of probiotics for the average individual, and we can start kind of like we can meander into more sports specific applications after that.
Yeah, for sure. There are a couple of, I think it was maybe in like around 2014, there are a couple really interesting studies that came out. One was a systematic review. I want to say like it might have been McFarlane et al., but they were speaking about the importance of looking at the strain level, and when we're doing something like a meta or a systematic review, that we can't just say like on the whole, like, oh, probiotics are effective or probiotics are ineffective, because the effects are really strain specific. And so we have to get down to that level of specificity when we're determining the potential efficacy of a probiotic supplement. And so when we're talking about strain specificity, we're really talking about like the difference between a dog and a dingo, or a dog and a wolf. So like you don't necessarily, like when you're on Craigslist and you're like looking for a puppy or something, like you want to know the difference. You're looking at the picture and you're like, I don't know, that's questionable. Is that like some kind of hybrid or something. And they're like, oh, like, don't worry, it's Canis, it's close enough. And you're like, no, I really need to know, like, are we looking at a dog or a wolf or a dingo? So that's what we mean when we're talking about like strain level specificity. Mm-hmm. And one of the biggest limitations that fortunately in recent years has been, it's becoming resolved. But one of our biggest limitations is that we can't easily gain that level of resolution when we are studying the microbiome, especially when we're doing like a fecal sample. Most of the time we're looking at what's called like a 16S RNA marker. It's a genetic marker, and we're able to look kind of like to the genus level, which is above species, and species is above strain.
Right.
So, we can see like differences in genera, and sometimes to the species level as well, but we're only looking at bacteria. So we're leaving out other microbes like archaea that actually seem to interact with bacteria quite a lot. Mm-hmm. And then like fungi. So there, we have yeast in the GI tract. And we also have to remember that we're looking at a whole ecosystem here. So just because we see an elevation in one group of microbes, that doesn't necessarily mean that like whatever our intervention was directly affected that group. It could have been that we affected some other microbe and then it produced some postbiotic that this group was able to use as an energy source. Yeah. So it gets, So it's like, it's much more complex than like, okay, I take this pill and then this level of bacteria is elevated in my poop, and then that means that it's in my gut. Because the other limitation is that a fecal sample does not actually represent the whole of the GI tract. Mm-hmm. It is really a surrogate for the distal part of the colon or the large intestine, but we're missing out on the small intestine and the parts of the large intestine that are closer to the small intestine. Mm-hmm. And we can look cross-sectionally too. We look at the lumen, the inside of the intestine, versus the mucus that's covering the intestinal cells. Mm-hmm. And we really don't get much mucus representation in a fecal sample. Mm-hmm. So we can't assume that if just because a group saw enrichment in their fecal sample, then that means that probiotic also enriched in their gut.
Right.
And that might not actually even matter. We don't know that matters either. So it could be that it's just a transient effect. They go through, they cause some improvement, and then they leave, and it's okay that has happened. Yeah. So keeping in mind all of those limitations, it looks like the most consistent applications that we have found benefit for would be that of reducing diarrhea. That would be diarrhea associated with traveling, so like kind of just like an infectious diarrhea, that associated with antibiotic use, and that associated with C. difficile infection. Okay. And right now it looks like one of the most effective microbes would be the yeast S. boulardii. Seems to be pretty helpful across the board. If you're experiencing diarrhea, then taking that will probably help at least a little bit. And then there are some strains of bifidobacteria and lactobacilli that also seem to improve the severity or duration. And then we see some applications as well in irritable bowel syndrome, mostly IBS-D, and that it can improve symptom scores, kind of just like global symptom and quality of life. And then VSL3, which is a multi-strain probiotic that is, depending on the type you get, can be quite potent. That seems to improve inflammatory bowel disease. I believe it reduces recurrence of Crohn's, or I want to say Crohn's. So there are some applications, but it is really kind of malady specific, and it is strain specific. So not all strains of bifido would help necessarily with, reducing diarrhea. Right. And it also seems to be that there could be some population specificity. So some people appear to be fairly resistant to enrichment with probiotics. Mm-hmm. There's one group that has actually gone through and sampled from like the mucosa, and they looked at the cecum, and they sampled all along the GI tract, and they found that some people didn't see enrichment with probiotics, although their fecal samples would have indicated that there was enrichment. So there was an elevation in fecal sample number of that probiotic bacteria, but not along the GI tract. So it's kind of interesting to see that. We don't know who might be resistant. It's just, it might be you. And so you could be, wasting your $50 a month or whatever it is on that supplement. One interesting example would be Lactobacillus rhamnosus GG. That's a specific strain that seems really effective for pediatric diarrhea and doesn't seem to do anything for adult diarrhea. So there could also be, the factor of, like it's because our microbiomes change with age. And so, and there's just like an individuality to it as well. So there's really no evidence right now that like everyone should be taking a probiotic or that there's sort of a kitchen sink probiotic. It really is specific to whatever your issue might be. And so for that, there's a really great website. There's AE ProBio and Probiotics. I want to say it's US Probiotics Guide. They have a whole compendium that they just updated this year on the applications of probiotics with a hierarchy of the level of evidence. So you can, check that out if there is something you're thinking like, I'm going to travel soon and maybe I'm interested in seeing what could help with that. That would probably be the place to go.
Okay, perfect. Along the ways, like, so when you mentioned that take a probiotic, it shows up in the stool sample, and then you don't really know sometimes, but is there a way that you can like tag or like biomark? You give somebody like a set quantity of the bacteria, and is there a way like we can like genetically modify them or tag them to where, all right, you ingest it, and then you collect the stool sample, so you've seen what's come out, and then you can kind of like take the difference and figure out like, maybe this much stayed in the colon or in the intestinal tract or anything like that. Is that possible, or is that just kind of am I just kind of talking out of the wrong end?
Well, no, I mean, we do that with, when we're looking at like doubly labeled water or, we give some sort of like radio labeled isotope to a person to
right. And that's what got me thinking about this.
Yeah. Like where did this carbon end up? So I imagine that it might be possible to give someone, a bacteria that has some sort of label to it. But then I wonder how we would ensure that label would then be transferred to, the sort of offspring, so to speak, of that initial group of bacteria. And also how we would then determine what the downstream effects would be in terms of like its effects on other microbes. So in some cases, what we find is the other thing we have to think about is like when we're studying the microbiome, not everything that we're looking at is really like quantitative. It's more qualitative. It's like looking at a teeter-totter, like this kind of goes up and this goes down. And so we see like a change in relative abundance, but we might not necessarily know like what the hard numbers are. And we're estimating a lot too, because there are different ways that we're analyzing that we're sort of like, okay, let's throw out anything that's like super low count. We'll say those don't matter, and so we're estimating a lot. So even if we were to count specifically, like these are the probiotics, these are the ones that we gave in the probiotic and they are in the gut right now, they're going to die really soon too. And so like that, that initial capsule that we take might not give us the whole picture because, they're going to be, gone in a couple of days. But I think that would be a really interesting way to see like what is actually going on, like who's actually staying, but what they generally just kind of look at fecal samples and they're like, okay, well, these are strains that we gave the people and these strains are higher now than they were 2 months ago. So it worked.
Perfect. One of the byproducts of the fermentation of fibers in the gut is butyrate. It's one of the short chain fatty acids. There's a few other ones that go there. And so butyrate's got a bunch of cool properties. It's anti-inflammatory, it's an HDAC inhibitor, and it's got some other really, really cool things that are going on. We see supplement companies coming out with, various butyrate things to cure leaky gut, improve inflammation in the body and all of that. Can you speak a little bit to, is supplemental butyrate actually effective or is it bioavailable? Is it actually going to confer any benefit? Do we need to worry about how much butyrate we're producing? Things along those lines.
It's so funny you bring that up because I was actually super interested in butyrate when I was in grad school because I was reading so much about it and I was like, wow, this is really interesting. Like this seems to be associated with like exercise performance, with, improved endurance, insulin sensitivity, appetite regulation. I was like, I think like, I'm going to try this. I'm going to go ahead and try and supplement some butyrate. Yeah. Well, let me tell you, butyrate smells like, it smells like vomit. It smells like nasty Parmesan cheese vomit. And I opened the bottle and I was like, whoa, like, oof, okay, that's rough. And my dad, he was really interested in it. So he wanted to buy some too. And I was like, yeah, sure. It's great stuff. Yeah, just try it and see what you think. And I didn't tell him, that it was going to smell like vom when he opened it. And he was like, oh my goodness. So that was my foray into supplemental butyrate. And this was, oh my gosh, this must have been 2010 or something.
Mm-hmm.
So forever ago. And it's really funny now to see that my initial inclinations, my initial intuition, seems to play out that, it's been kind of a constant in the literature that butyrate is associated with all of these, beneficial health outcomes. Mm-hmm. Now, as far as actual supplementation with butyrate or propionate, which is the other one that's kind of recently come into the spotlight after looking at like the guts of marathon runners and Veillonella and how it's producing that, in rodents seems super effective. Actually, supplementing with propionate actually has improved exercise performance in rodents. But in humans, I think it's sort of the same difference in the magnitude of effects that we see with most supplements. Yeah. And I don't think that it would be as prudent to try and supplement with butyrate or propionate as it would be to spend that money on a variety of plants in your diet, so that you're providing, the substrate to support both microbial diversity and the production of those short chain fatty acids. And the short chain fatty acids are really made in sort of a specific ratio, or found generally in kind of a specific ratio in the GI tract. And that certainly could be something to consider, that it's not necessarily better always to have more of something. Mm-hmm. So, would we potentially, if we were supplementing with butyrate, would we potentially change the pH and the fatty acid environment of the GI tract in a way that would influence the microbiota? Because that can happen, too, with when we're looking at changes in like secondary bile acid metabolism and whatnot, that sometimes just the changes in the pH in the colon can have an effect on the microbiome. So that's so far, and it's been a while since I've looked at like the actual exercise performance literature. But again, it's like, I don't know that I saw anything that was like super compelling in humans.
Gotcha. All right. Along those lines, with the composition of the gut microbiome, so there's a couple of factors that play into it, like sleep, stress can affect it, your diet can affect it, genetics play a huge role in it, obviously, like it dictates a bunch of other stuff in our life. How much control do we have on a day-to-day or meal-by-meal basis over like the composition, function, structure, health of the microbiome?
It's really interesting that you bring up genetics, actually, because that has probably the smallest influence on the residents, on the resident microbes. It's estimated as about maybe a little over 10%. So our genes would explain maybe about 10% of the microbial community that we would find in me versus you. And there are some uncontrollable aspects that seem to also have a more significant impact. So maybe about 30% of the individual variability that we see between individuals would be explained by a combination of their age, their gender, and their geographic location. Okay. And so when we're young, we look we have a kind of a young person's profile. After we introduce solid foods, it starts to stabilize and remains very stable over the course of an adult life until we get to about age 60 or so. And then we start to see sort of a reduction in the diversity. It kind of goes to a less mature state, similar to what we saw, in infants. And then there may be some gender specific differences as well. We do see this in rodent studies quite often, that dietary effects may be different in male versus female rodents. And there are some taxa that are more abundant in females than there are in males. There are effects of estrogens on the microbiome as well. So we can take that in consideration. We can't do anything about those two factors. And then our geographic location, what's really interesting with that is that we see differences in the taxa. So we see differences in who's there, but there's a functional redundance. So even though the members, like the employees are different, they're doing the same things, you know? So it's like this in the UK, you know, it's John, and over here it's Jared, but they're both cashiers. And so there's that functional redundance, and that's really helpful for us because one thing that people may not realize is the extent to which we offload a lot of metabolic processes to our microbes, and they're actually harvesting, you know, about maybe 10 to 20%, depending on the amount of fiber that we're taking in. They might harvest about 10 to 20% of our dietary energy for us that we don't have access to because we don't have those digestive enzymes.
Yeah.
And then the rest of it, maybe about 20 to 60% of the variability is explained by our lifestyle. So our dietary patterns and our physical activity habits, our medications, and you know, perhaps like the presence of the presence of a disease is probably even lower than that, but it's a huge range. I mean, it's 20 to 60%. You know, we're talking about like a very small portion versus over half. And it's just that we don't know because, you know, when we're there's a really kind of like a landmark study looking at the relationship between diversity and fitness would be out of Estocchi's lab. And they found that that cardiovascular fitness explained about 20% of variations in microbial colonies between people who are fit versus people who are less fit or who are sedentary.
Yeah.
But, you know, in our dietary pattern, I've seen estimates as low as it explains maybe 15% of the variability, you know, all the way up to if we combine diet and lifestyle or diet and physical activity, then maybe we can account for over half of that. And then there's the kind of like the foundational core of our microbiome that is just human species specific. Mm-hmm. So you are about one third of your microbes are just like you because you're human, and then the other 2/3 are influenced by all these other factors.
Fascinating. I didn't realize. Like I knew of a few of those factors, but some of those other things that's pretty cool. Interesting. Mm-hmm. How dynamic are the changes like over the course of a day? So say, you know, normally you're eating like a normal healthy lifestyle, but we all have those days whether we're traveling or something, and like the only thing you subsist on is a Subway sandwich at the airport and like 6 protein bars. So how big of an impact, you know, how big of an earthquake is that to your microbiome? And or is it, you know, is it a little bit more robust and resilient than that to where like one day of getting thrown off your A game of eating isn't really that disruptive?
I like that you said earthquake because yeah, I mean, we're thinking, you know, like the magnitude of effect for those guys. They're like, this is all of our food is gone. So there was another landmark study that was done I think out of like the Gordon and Turnbaugh lab. And these are names like if you want to go back to like the foundational, you know, how did we ever link like obesity to the microbiome? It's out of this lab. And they this was actually a human study. So they had one group that switched to what we consider to be a vegan diet, and another group that switched to just an animal-based diet. So it was meat and cheese versus vegan diet. And the people who went from their, you know, normal omnivorous sort of like westernized diet, because I believe this is in the UK, it might have been the US, but it's like a westernized diet. They switched to vegan and really didn't see much of a change from one day to the next. The people who went to the animal-based diet saw significant changes within 24 hours. So they saw some big increases in Bacteroides. So this is a group that they're known to be mucin degraders, and they're really adaptable. Like they don't mind if you don't have any carbohydrates in your diet at all. They're like, oh, that's fine. We'll just like get them from the mucus layer, or we'll just metabolize amino acids, whatever. Whereas bifidobacteria, it's a group that is really finicky and like they really need a microbe with accessible carbohydrates. And if you don't have any, they're just gonna they're leaving. They're not gonna stick around, you know? And what's really interesting about this is that I have heard some proponents of like the carnivore diet talk about the increase in beta diversity in this study. Mm-hmm. And maybe thinking that beta diversity means an increase in diversity. And we have associated diversity with gut health. We're like, oh, this is a good thing, right? An increase in diversity. But really, that's a misunderstanding of what beta diversity actually is. Mm-hmm. So alpha diversity, that actually refers to the richness and evenness in microbes in one sample. So that's how many different species do we have there and how evenly are they represented? And so we want to see a lot of different species. We want to see that, like I mentioned earlier, that we have kind of a higher relative abundance of the less pathogenic or non-pathogenic ones. But we don't want to see that anyone has that we would expect to see there is just not there. Like we're going to have some potential pathogens. That's normal. So that's alpha diversity. Okay. And one person could have, low alpha diversity here, and then they increase their fiber intake. And then 6 months later, they have high alpha diversity because they have greater richness and greater evenness. Beta diversity is actually referring to the similarity or dissimilarity of two samples. When we have high beta diversity, it doesn't mean that the sample is rich and even. It just means that it's very different from the other sample. So what we were referring to with beta diversity is just, wow, the people who went on the meat-only diet were really different after they did that versus before. And the people who went to the vegan diet were not very different after versus before. But we don't have any information about the actual alpha diversity. So that's something that I think got really conflated. That message got conflated, what that study was actually saying. And they actually brought up some concerns that, these Bacteroides, because they're reducing that mucus layer thickness, they're actually reducing one aspect of our immune system. That's pathogen control. Yeah. And the other concern is that as they are metabolizing amino acids, yes, they can ferment some amino acids to short chain fatty acids and branch chain fatty acids, but they're also producing some things that are associated with colorectal cancer like indoles and sulfates and some things that we probably don't want to have accumulating in the GI tract. Now, if you do that for one day and then you go back to your normal habitual diet, then it will rebound, because that's what they saw in the study too. They're like, okay, and we have people go back to their habitual diet and then it was back to normal very quickly. And we do see a circadian rhythm as well. So you're fasting overnight, and so you will have lower abundance of microbes that need food right now. Like some of them are not going to make it through that 12-hour fast. And then in the morning, you start to eat, and then you'll see a bloom in those that are responding to that food. And so there is a circadian rhythm. So you'll see, even if you were to do, nothing, you would still see a fluctuation over the course of the day. But even that, when we're looking at Bacteroides, when we're looking at just one genus that may have, a few hundred species or something, there are potentially thousands of species that we have, because we haven't been able to identify. We don't know everyone that's there yet. Right. But there are hundreds and hundreds of species. So if we see, just a change in one group, what does that actually mean, as a whole? And if we go really high, sometimes people will look at the phylum level, Firmicutes versus Bacteroidetes, and they might see some changes in those. But that's looking at vertebrates versus invertebrates. Because it's telling you nothing about what's actually going on, functionally. Yeah. And the functional aspect is one that we kind of miss out on too, because like I mentioned earlier, just because who's there changes, that doesn't necessarily mean that the function changes. And so if we look at gene activity, it might be, it doesn't really matter. So over the course of 5 years, that's about as long as a study has looked longitudinally at changes in the microbiome. Looks like about 60 to 80% of the microbes present are consistent that whole time. And then we might have something like a dietary change, in 24 hours, or we take antibiotics and maybe we see a perturbation for a couple of weeks, maybe up to 6 months. Maybe we get to a new stable state. Maybe we don't keep, that we're not unhealthy just because we have a different profile over time. Correct. It's just, keeping in mind that we probably have relatively little control over who's there. And ideally it's a resilient community. So it kind of can bounce back after there's been some sort of perturbation. I hope that gives people a little bit of peace of mind that you don't have to do anything super extreme, but it's probably not a good idea to do something super extreme. If we're doing the carnivore diet, when we're completely eradicating all sources of nutrient for many of those microbes, that's probably not the most prudent way to support, gut health.
Since you brought up the carnivore diet, one of the things that, the guys on the extreme fringe of that, and the same in kind of the keto community, the paleo community, primal community, there's this fear mongering that's going on around plants, fibers, and anti-nutrients. Now, I don't want the listeners to make a mistake. Anti-nutrients are a real thing. Like there's oxalates, there's phytates, all these kind of things. But I've always been of the opinion, the degree to which these things, unless you're like only eating 38 pounds of spinach every meal 5 times a day, you really don't need to worry about that. So can you speak a little bit to, what anti-nutrients are, ways to mitigate any potential risks from them? Do we even need to be concerned about this? Or is this just, these unscrupulous individuals preying on the uninformed?
Well, I would agree with the lack of scruples in people who are, fear mongering about like fruits and vegetables and legumes and whole grains. Because like you said, it's really about the dose, and in a prudent omnivorous diet, the amount of anti-nutrients that we could get, when we're looking at like phytates and oxalates, is incredibly low. Like, and even if it's in a measurable amount, what we do and what we have done with these foods for centuries is that, we soak them or we cook them. And that either changes sort of like it changes the chemical composition of those, or it helps to break them down, or they leach out into the water. And so just like we don't eat, raw potatoes, so like it's little examples like that show that humans have found ways to include these foods in their diets for many decades, hundreds of years, probably by trial and error. Maybe at some point someone did eat a bunch of like potato greens and then they died. And then people were like, let's not do that. That's not a thing that you want to eat. Okay, that's bad for you. Yeah. But yes, now in this day and age, I think people maybe are kind of like trying to look for something to worry about, like, lectins. And sure, if you put something on a petri dish, if you apply something to cells, you could dose it enough that you would cause an inflammatory response. And you can do that in, perhaps even in a mouse model. There are mouse models of colitis that, we can feed things to, and then it will exacerbate their colitis. But, no researcher would be like, yes, that you extrapolate this directly to humans in this exact dose. No one's doing that. They're looking at that. That's a mechanistic study to see like how does this happen at a cellular level? Because you can kill those rodents and take their intestines out. You don't do that to people. So yeah. And, looking at the abundance of evidence illustrating the benefits of including fruits and vegetables and whole grains in one's diet, it sort of boggles the mind that people would be like, well, look at this one study on diverticulitis and diverticulosis. And, sure, yes, there are some studies that have shown that like there's not a clear causative link between, taking in fiber and it like reducing diverticulitis. But that doesn't mean that fiber causes diverticulitis, or that not having it, or that you don't need it.
Right.
That's you cannot conclude those things from that type of study. Or, like the example with beta diversity, just kind of an incomplete understanding of what that actually means, and using that to promote your ideology, I think is potentially harmful.
Yeah, and another, like, kind of building off of that, and I guess taking it on a little bit of separate of a tangent, is another argument that they'll say is, and I'm saying like they as in like the collective people that kind of prey on all these little insecurities of the mass populace, is that we haven't evolved to eat certain foods, like those other fillers, stabilizers, additives, artificial sweeteners that are in your protein bar, your pre-workout, or whatever other little packaged food you get at the store. I'm not saying those should be the base of your diet, but they're saying, well, we haven't evolved. We've evolved with strawberries for thousands of years and red meat. We haven't evolved with these foods, so our food bodies don't know what to do with these things. Is there any truth to that? Can you kind of explain where they may be truthful, or where maybe they're misleading somebody?
Well, I mean, we haven't evolved to produce digestive enzymes to break down fiber either, and so like there's truth to that statement, in that we have not evolved really to eat any specific food or not. but we have evolved with a set of digestive enzymes and a digestive process that can break down most things. And the things that we don't break down are either metabolized by the resident microbes, much to our benefit, or we excrete. And so we can measure our metabolism of these products, and we can measure the excretion in feces and urine, and determine like which, what percent comes out and what percent stays in, and like what's metabolized. And the things that, come that reach our tables have been through a long and arduous process of testing for human health and safety. And I have seen one recent review looking at potential effects of food additives on the gut microbiome, because that is, to be transparent, an area that has not really been included in studies looking at the effects of food additives. But because we don't have the data, we really can't make conclusive statements in either direction to say like, yes, 100% this is totally safe, or like, no, this is probably super dangerous, it kills all your microbes. But we have to be very careful in extrapolating from like cell culture or rodent studies. And that quite often is the case, especially when we're looking at something like artificial sweeteners, and people say, oh my gosh, it's going to like kill all your microbiome. And in fact, when we look at the human data, which is limited still, we just don't see that that's the case.
I want to go touch on, since we brought up digestive enzymes, that was one of the things I wanted to touch on with you. Very often we'll see digestive enzymes added to protein powders or some other kinds of, gut health supplements. And I've always wondered, are they actually having any benefit? Like, because our bodies are going to produce the digestive enzymes that we need. Are these enzymes actually just getting broken down and eaten alive by the stomach acid that's in our stomach? Does it survive that and make it into the intestinal tract to help break down these foods and facilitate absorption and utilization to a higher degree? Or is it really just kind of window dressing on the labels?
Yeah, for the most part, they're not going to do anything at all, especially if they're not, if it's like in a powdered form and it's not enterically coated, they're going to be broken down just like under any other peptide would. Once it hits the stomach acid, it's unfolded, and then an enzyme would lose its functionality once it loses its unique shape.
Right.
The exception to that would be lactase. So the lactase enzyme does seem to help reduce some of the GI distress we would experience if we have lactose intolerance. But you have to have a fairly small amount of lactose and a fairly large amount of the enzyme. And the same can be said for beta-galactosidase, which is sold as like Beano. That also seems to be pretty effective in helping people, with gas that might be associated with like beans, for example. So, and those are often sold as, they're either enterically coated or they're packaged in some way that they would survive transit, through the stomach. But yeah, the rest of it, the things that we would get, there are a lot of digestive enzymes that are, produced by like, they're extracted like things that are produced by like insects or something, or that we get them from fruit. And when you look at the pH range in which they're active, they would not survive transit through the stomach. Okay. Now, if they're enterically coated, maybe, but even then, what are they supposed to be acting on in the gut? So like enzymes aren't just, it's not that we're just dissolving things. Like enzymes have a specific action and they have to like fit into the lock and key thing and whatnot. So yeah, it's not sort of just like a willy-nilly, like you're pouring acid on it and then it's dissolving. So yeah, so those have a limited application, and it's probably easier to just get like a lactose-free version of something if you can.
Yeah, those are part of the reason I asked, because like we'll see protein powders will have like this comprehensive digestive enzyme blend, or like a glucose disposal agent where you're supposed to use a cheat meal, so it'll have all kinds of lipase, cellulase, proteases, pepsin or papain, and all these other kind of enzymes. I'm thinking, are these actually doing anything? Like from a mechanistic standpoint, I understand what they're supposed to be doing, but I've always been of the opinion like the stomach is a super acidic environment. I've got to understand it's probably breaking down those enzymes into individual aminos, like you were saying.
Yeah, that's exactly it. I mean, we don't even, we have salivary amylase, and that's inactivated as soon as it hit. Well, if you make salivary amylase, so that is denatured once it hits stomach acid. And I think lingual lipase also is denatured when it hits stomach acid. So for the most part, like your stomach acid is there to unfold all the proteins, and then the fats and the carbohydrates wait until they get to the small intestine, and then you produce, you know, a wide variety of digestive enzymes to handle that. And, you know, with the carbohydrates that contain the linkages that we can't break down, they reach the large intestine, and then they're metabolized by the bacteria. So they're kind of doing that for us. And yeah, it can be super uncomfortable to like experience gas, and that can be managed to some extent through the amount of the kind of gas producing vegetables that we're taking in at one time and over the course of the day as well.
Gotcha. Green supplements. So in addition to digestive enzymes, green supplements is another thing that's been a huge trend in the supplement industry as of late. I'll reserve my opinion on it for a second. I want to get your opinion on it. Like, so I don't want to speak out of turn on anything. So what do you use them? Do you find any utility in them? What can we expect? Is this just kind of like another cash grab for the industry, going for like that low hanging fruit?
Yeah, it's like, this one's so interesting to me because greens powders are pretty expensive. And some folks are like, well, I want to use my greens powders rather than, you know, buying fruits and vegetables, because fruits and vegetables are so expensive. And I think, you know, there's like an expense weighed against maybe the perishability of those foods. And so like the greens powders seem less perishable. And so it seems like a better investment than spending like the $30 on fruits and vegetables and eating those. So the problem with greens powders is that, you know, quite often we're looking at a proprietary blend. So we don't know like the relative amounts of what's in there. And it's usually a blend of, there might be some like herbs and various grasses and some fruit and vegetable extracts, digestive enzymes. Sometimes they'll put in like a fiber supplement. They'll put in a probiotic. So it's just this whole like conglomeration of things. And people are like, it's equivalent to however many servings of vegetables. And, you know, when I've asked, how are you calculating that? You know, I think they're going by sort of like the vitamins and minerals maybe, and like the antioxidant capacity of that food. Not to say that antioxidant capacity is actually like physiologically relevant, but that might be what they're looking at.
Right.
And then, you know, the micronutrients available. So maybe if we were to take just like the food, like just the framework of like, okay, take this vegetable apart and just break it down to like, it has this much vitamin C and this much vitamin A and vitamin K. It has this many grams of fiber. It has this much water. Just get rid of the water. We're going to dehydrate it. And then we're going to powder that and put it into a capsule. And it's like celery and broccoli and whatnot. Then perhaps because those micronutrients can be fairly stable when dehydrated, perhaps if you ate enough of that to equate whatever your 5 servings are, and I don't know how we would quantify that, but you'd have to just be like, I dehydrated 5 servings and it made like a cup of powder, and then you ingested that. Like maybe that would be feasible. Yeah. But when you have like someone else doing that in a lab and you don't know, you know, how they've created it, and not to be chemophobic or anything, but like it's a proprietary blend. So we don't actually know what's in there. And, you know, you're paying for some things that are probably not useful, like the digestive enzymes and superfood blends. That's a complete nonsense made up term. And, you know, I've seen one picture where like the person had taken out, you know, however many milligrams of like superfood blend and really like put it on their fingertip and like to show, you know, very tangibly, this is the amount of superfood that you're getting in a serving of this. Like, you know, like why not eat the fruits and vegetables and whatnot? Now, in terms of looking at the data, because I don't want it to be just my opinion, the problem is that we don't really have studies on greens powders. Like no one has been like, we're going to give these people this greens powder and see what happens. What usually we find is that there are a couple of companies, like MLM style companies, that often partner with medical practitioners to sell a powdered blend of fruits and vegetables. And then they look at some health biomarkers, like they might look at blood pressure, or they might look at, you know, markers of like DNA methylation or something. And they may find that there is a change over time and then say, okay, it was definitely this product. But the design of those studies is often questionable. Like they might not have a control group, it might not be blinded. It might be that they're studying something, they're looking at a marker that has no physiological relevance, like this doesn't actually matter, or it's within such a huge range of this. All of this is normal. So you went from like this end of normal to this end of normal. So the empirical evidence for using those and like having some benefit on your health is lacking. Mm-hmm. And the evidence for eating fruits and vegetables is huge. So like, I would say, if you got that $30 or whatever, maybe more, I don't even know, to spend on the greens powders, maybe consider how you might be able to invest that in like whole foods that are also more filling as well, So like, that's my take on it.
Yeah, and that's basically what I was thinking, and whatever my response has been. When they come out, it's 46 ingredients in a 2.5 gram blend. I'm thinking, all right, 50 milligrams of kale powder, 50 milligrams of broccoli powder, and then we from the consumer aspect, you have no idea the processing means. I mean, sure, if we're going to be charitable, we'll say, all right, they use the cleanest, cold pressed, freeze dried way to preserve as much micronutrients as possible. Mm-hmm. But you still have there's no micronutrient profile listed on any of these supplements because the contract manufacturers probably have no freaking clue on what's actually in these things aside from like the little green dust that's in there.
Yeah, exactly. And I'm like, well, you could take like a multivitamin, like if you want to have like an extra kind of like insurance policy, and if you're a small female and you have a fairly low caloric intake, then it might be prudent to go ahead and like take a multivitamin or at least just supplement with the things that you would be deficient in. Like if you're not eating dairy, if you're not eating red meat, then you might look into some calcium, vitamin D, maybe some iron. But like, it's just like when we look at the probiotics and people are like, more is better, right? Mm-hmm. No, not necessarily. Like more is just more. More is more expensive.
Exactly. I'm glad you brought up the multivitamin thing. That's another, kind of kickback where we get like somebody will kick something and say, well, no multivitamin tablets are good. Basically, it just goes in and out of your system. I'm thinking, have you ever pooped out an entire multivitamin tablet? I'm pretty sure that would hurt. I don't think it's so. Can you speak or touch on a little bit of our body's ability to break down these multivitamins, whether it's in a capsule form, which is probably going to be easier to break down versus a compressed tablet, like you would get like your Flintstones multi or a Centrum or something like that?
Oh man, Flintstones! I would just eat those for fun.
I would too.
Yeah. So from what I've seen, the bioavailability of most multivitamin supplements would be around like 50 to 60%. So so the recommendation is just to get one that's got like 100% of your RDA, your DB, and then take that, and then also eat a prudent diet, and you'll probably make up for like the other 50%. So water soluble vitamins, you do indeed, like you have an excess, you'll lose those in your urine. But with a fat soluble vitamin, so A, D, E, and K, you can actually store those in your body fat. And so with those, you could potentially experience a toxicity if you were to take sort of like super doses. So sometimes people are like vitamin A for like, antioxidant, vitamin K could potentially like interfere with blood clotting medications. So supplementing is a concern more so with the fat soluble vitamins. Right. With water soluble vitamins, you're probably like if you take too much vitamin C, like you'll give yourself diarrhea, and it's not going to get you over your cold faster anyway. So with some of those things, it is a little bit wasteful. Mm-hmm. And what also could influence the bioavailability is if you're taking minerals, some of those will carry a charge, and so if you're and they will kind of compete with each other for absorption. So I think I've seen with like, maybe I want to say it's like zinc and iron and copper interact with one another in such a way that like if you take a lot of one, then you might not, absorb the others. So keeping those things into consideration, then, you might want to like take them on alternating days. Mm-hmm. But the other thing is, these are vitamins and minerals are not destroyed like macronutrients are. Like we break those down and release energy. But when we're looking at a micronutrient, a vitamin or a mineral, it's going to either be used directly or in concert with some enzyme to catalyze metabolic reactions. But we don't destroy that. It's just, it's recycled. And so sometimes we may have like a dietary deficiency for a little while, and we don't actually reach a clinical deficiency like in our bodies. So even with a multivitamin, you probably don't have to take that every day. You'd probably be okay taking it like every other day, maybe even every week, because like we're just kind of maintaining that along with a prudent diet.
Okay. I want to go back to the sports nutrition and probiotics concept. We touched on that, and they've done some links, like endurance athletes have a different kind of makeup, or like you could, like, endurance exercise, since it's so strenuous, can negatively impact the microbiome. And can we, I guess, let's discuss, the effects of probiotic supplementation on just regular training, endurance training, and then is this going to be to a point where we're going to start seeing like probiotics slipped into our pre-workout, because that's going to give us help us lift like 10 extra reps on the bench press or something like that? So can we delve into that world?
I can sum that up very quickly, because there's no effect of probiotic supplementation on performance. Like, it's been studied a lot. Like, I know people really wanted that to happen. Like, they wanted it to be a thing. I don't know if people have seen Mean Girls, I think, but it's like, stop trying to make fetch happen. Like, stop trying to so far, like, that is not going to be a thing that's going to happen. So it was really, there was actually, I think it was in an ISSN article, they mentioned that they were looking at, I think they were comparing groups of swimmers. And I want to say that they had they had two groups of swimmers. There was no difference in performance in the people that got the probiotics versus those that didn't. So no improvement in performance. But the group that had gotten probiotics had like a slightly higher training volume. And I was like, I don't think that this is a good thing. You have higher training volume, but you didn't see an improvement in performance. Right. So I don't think that like maybe probiotics were like a bad idea. But that's just, I'm just extrapolating from that. But yeah, so we really haven't found that they've been beneficial to support exercise performance. But they may play a role in enhancing the energy availability of our diets in a way that could support our exercise performance. So we can think of exercise, diet, and microbiome as this sort of triad. And there's bidirectional effect in every direction. So obviously, our diet supports our training volume. So we need to have adequate carbohydrates, protein for recovery, fats for adequate energy intake. And the more training volume that we accumulate, the better our performance, usually, to a point. And then also our level of cardiovascular fitness is associated with increased microbial diversity. And so is the diversity and the adequacy of fiber in the diet, also associated with microbial diversity. Yeah. And there are some microbes that have been associated with either the production of glucose or the clearance of lactate and the production of propionate, which is a short-chain fatty acid. And those microbes have been enriched or they're in higher numbers in the guts of elite endurance athletes. But we don't know that those microbes cause them to be elite, or whether it's that their training volume increases the abundance of the substrates that they need. Like these microbes need lactate. So if we accumulate lactate, we feed them, and then they make propionate, and then propionate feeds us. And it's a nice little beneficial cycle there. But supplementing with probiotics doesn't confer any additional benefit. It's just like what we have is what we've got going on is great. And so, but we don't know we don't really have that elucidated in resistance trained athletes yet. But what we do see that's really interesting, this was a recent study. So this group has done a couple of studies over the course of the last several years. The first one was just observational. They were just looking at bodybuilders and cross-country runners and finding correlations between characteristics of their microbiomes and their diet. And they found that really high protein intake or really high fat intake at the expense of dietary fiber was associated with reduced diversity and reduced bifidobacteria, respectively. And so bodybuilders were eating 100 grams of fat a day and eating 15 grams of fiber a day. So fast forward a few years, they took a group of bodybuilders and gave half of them a probiotic, and then the other half no probiotic. And then they also had a group of sedentary controls. And what they found was that the probiotic did nothing, but the bodybuilders who were ingesting inadequate fiber, their microbiomes were not significantly different from the sedentary controls. Whereas those that were ingesting adequate fiber, they saw what we would expect to find, based on what we know now, an elevated level of diversity because they're physically active people.
Right.
So probably our dietary fiber intake, our dietary pattern could be mediating the relationship between exercise and microbial diversity. So even if we do the exercise, if we're not feeding them with adequate substrate, we're not giving them the energy that they need, we may not fully realize microbial diversity. And this was something that I had kind of, I don't know, dreamed up like back in about, I think it was like in 2017, I was just starting to present on the relationship between the microbiome and exercise. And I found that exercise is quite often associated with specifically elevations in levels of butyrate producers. And that's kind of like getting back to, what we were talking like early on, people are like, oh, like butyrate seems to be a good thing. Yeah. And so I thought, well, this is really interesting, but these really do need microbe accessible carbohydrates. Like we're not going to get this abundance of butyrate, fermenting amino acids. And there's really no fat metabolism in the gut. So like carbohydrates are probably really important. And I said, I think that like we probably need to make sure we're having a prudent diet. And it was cool to see that 4 years later, this study was like, yeah, taking a probiotic probably doesn't do anything anyway, but also you need to be eating a prudent diet.
Yeah. Fascinating. Awesome. A common complaint with creatine is that a lot of people suffer GI distress from it. Is there, and what I've said maybe is, hey, maybe take it with a meal or maybe break your dose up during the day. Like don't take all 5 grams at once, maybe split it up, morning and night or morning and lunch or something. Is there anything else from like a mechanistic standpoint, why certain individuals seem to be more prone to GI distress with creatine than others? And then is there any steps you would have, you would recommend they do as far as divide the dose, take it with certain foods, don't take it with certain foods or something, or maybe like enzymes, probiotics, whatever, anything like that?
Well, I think, a lot of it probably was like carried down from the old loading protocols where people would take like 20 grams of it a day for like 5 days and then taper down. And thank goodness we know you don't have to do that anymore. So, you can probably, if you're omnivorous, you can probably get away with like 3 grams per day. And that might be a little less troublesome. But really, it's most likely that it's just because it's an osmolyte, because it attracts water, that there could be a sensation of like pulling water into the GI tract and like some loose stools and things like that. But what I've found is that there's not really like kind of the sensation of like bloating, or like retaining water like subcutaneously under the skin. That doesn't actually seem to occur. It does cause intramuscular water retention, which is what we would want as part of its kind of mechanism. But yeah, I would say, probably just taking it in a smaller dose, like aim for 3 grams or so. And, taking in adequate fiber, like mixed varieties of fiber. So if we get soluble fiber, that can help to absorb some of that water. It creates like a gel and that can move things through. So it might actually help to take it with a meal and not like right around training. So you don't actually have to take creatine like right before you train. You can take it any time of day. So if it feels more comfortable to have it, with a meal earlier on or later in the day, that could probably mitigate some of the GI distress.
Perfect. Can you touch a little bit on what your own, diet supplementation training is, if any?
Well, I would say that I take supplements. So I do take creatine, and beetroot powder and citrulline, as, supported enough vasodilators. And I studied citrulline when I was working with undergrad still. And so I think it's just kind of like a near and dear to my heart. I know the effects are not super significant, but when you get like a whole bag of it and then like a year later, it's like, this probably isn't super effective. It's like, well, I'm just going to finish it anyway. But yeah, I take creatine every day. I'm trying to think of what else. I just kind of like, caffeine. That's probably the most effective supplement that there is. I do plenty of caffeine.
Do you have a preferred delivery form of caffeine? Coffee, energy drinks, pre-workout, anything in particular?
I don't do a lot of pre-workout because I find that the combination of like citrulline and beta-alanine and caffeine is like really rough on my stomach.
Yeah.
So my caffeine is in the form of mostly like coffee and like the little like caffeinated like powder water things.
Yeah.
And so plenty of caffeine. I love it. And I'm trying to think of what else I take, but yeah, I would say those are my primary supplements. In terms of like dietary pattern, it is what I would probably closest to the Mediterranean dietary pattern. So because I like cows and pigs, I don't eat red meat. I don't eat pork. But I eat eggs, and I don't really like chicken. I just don't like chicken, so I'm kind of like, take it or leave it. But I'll do like eggs and egg whites, lactose-free dairy because I'm lactose intolerant, a little bit of fermented dairy because the evidence for that is pretty compelling that it's supportive for just in health in general. And yeah, I eat, plants at every meal. So plenty of oatmeal, barley, whole grains, fruits, veggies, fish, and most of my fats come from salmon, and then like walnuts, chia seeds, flax, peanut butter, like one of my favorite things. Peanut butter is not like a super great source of omega-3 fatty acids, but I love it anyway. So yeah, that's kind of it. And I just don't count macros or anything anymore. I just, when I get hungry, I put stuff in a bowl and then, don't eat with distractions. And then when I'm done, I'm done. So that's kind of how I go about it. And I mean, I've been tracking macros for, since I was 19. So at this point I can eyeball things and I just have a general sense of like, this stuff tides me over really well. I know I'm going to be, really satisfied with this amount of food in this mix. And like right before a workout, I'm not going to have a bunch of like fiber and fat and whatnot, because that's going to upset my GI tract. But yeah, that's basically it. Like, not the most technical thing. I don't take probiotics all the time. When I'm traveling, I'll take Butyrate sort of like, as a preventative measure. So I started about a week before traveling. I take it through the duration of my travel and then about a week after, just for kind of like protection for, traveler's diarrhea. And I took it the last time I was on antibiotics. So I had some facial surgery stuff and I was on antibiotics for like 2 weeks. And I took Butyrate the whole time and I had no issues. And so, I do it like as, with its specific applications. But yeah, that's about it. That's my supplementation routine. I like take iron. I take an iron tablet like once a month because I don't eat red meat, and I take calcium. That's the other thing.
Awesome. Yeah, I think that's all my questions. I would like to dive into the health coaching stuff, but I think I'll save that for because that's such a giant other topic to tackle.
Yeah.
I mean, we'd spend another 2 hours probably delving in all that and like the pitfalls with that, because more people are doing coaching these days than ever online, and somebody that's been in the game for as long as you have, you can probably identify like, yes, get into this, maybe watch out for this. These are some good tips to abide by. Maybe we can schedule that for a future podcast in the coming weeks or so. But this was a blast, Dr. Fundaro. Thank you so much for the time today.
Oh, you're very welcome.
If you have anything you want to plug unabashedly and unashamedly, feel free to go right now.
All right, sure. I appreciate that. So yeah, on the topic of coaching, my colleague Shannon and I are putting on the second iteration of our webinar series to talk about the comprehensive coaching framework and bridging the gap between sort of the dissenting opinions in the field. So there's people that are like pro-diet and anti-diet, and we kind of try to work in the middle and say, well, here's all the data, and like whatever you want to do with it is whatever you want to do with it. So we talk about those kind of contentious issues, and then some, how we can best guide our clients like on their side. We don't necessarily have to pick the side of the debate. We pick the side of the client, and we ensure that we are conscientious. We realize our responsibility to them, and that we collaborate with them and form a partnership to help guide them to their place of flourishing health. So that's when they're feeling like they're living a really abundant, authentic life. And they can check that out at btgcomprehensivecoaching.com. I am in the midst of updating my website, but you can find me at on IG at Vitamin PhD, and that's where you can learn more about coaching and the gut microbiome. At the end of this year, fingers crossed, Renaissance Periodization will be publishing the Gut Health Science book. So that is my review and application of the literature, so people can navigate the marketing, in an informed way. And I do provide consults for GI distress, for people who want to learn about probiotics, for people who are interested in sport nutrition, and also for people who are working on establishing a satisfying relationship with food and body image.
Perfect. Awesome. Thank you very much. I'm going to include links to all of that in the show notes. And thank you very much for your time. This was a wonderfully engaging conversation. Lots of good nuggets for the listeners. So I know they'll appreciate it too. So thank you, and have a great day.
You're very welcome.